All right, Becky, if you want to put up your last minute poll right before we start.
Shall I go first or second?
We're gonna have you go first. We've just we've been doing sort of like the more junior person going first.
Okay.
If that's okay with you, but we can switch.
Yeah, whatever is good for you is good.
It's all right, I'm screen sharing now.
So for anyone watching now, we have this really important poll that we want you to take. Go to menti.com and use the code on the screen. This is for my curiosity, so please weigh in.
I think you're wrong, Jan, in your hypothesis.
I am a little bit upset right now.
I think that dogs have been attending our seminars and have hijacked the vote.
Did dogs vote in this?
I really would have thought we were more cats.
Me too. It's funny because in all the other votes, you know, things kind of made sense, right? Favorite protein, ecadherin, right?
Yeah.
But this is just,
But this, a betrayal.
yeah. Makes
Can we
no
get
sense.
started?
Oh.
Adam, we've got some important data right here. It'll stop me. God, that hurt.
Cool. Mattia, do you want to share your screen in the meantime? Okay, so we'll get started. Hello, everyone. Another week, another seminar. Same thing as always, which is if you want to ask questions, just type it in the Zoom chat or the YouTube chat. And if you'd like to ask it yourself, just write the word question in the Zoom chat, and then we can call you to turn on your microphone. So we've got two speakers today. Our first speaker is Mattia Serra, who got his PhD in nonlinear dynamics at ETH Zurich and is currently a Schmidt Science Fellow in Applied Mathematics at Harvard. He develops data-driven models and methods and mathematical models to study complex biological and physical systems. And these systems are typically nonlinear, multiscale, and chaotic, and thus require new ideas to best uncover the underlying causal mechanisms and predict their behavior from the essential driving processes. And today he's going to be talking about dynamic morphoskeletons in development. Mattia, you have to unmute.
Yeah, sorry. Okay, can you see the screen and hear me now?
We can. You're in the slide view, not the presenter view.
Mm-hmm. Yeah. What about now?
Looks great.
Okay. So shall I start?
Yep,
Okay.
it was yours.
Okay, so first of all, thanks for attending the seminar, and also most importantly, thanks to the organizer for giving me this opportunity. This is basically joint work with Sebastian Strachan at UCSB and the Diger Lab at University of Dundee, who basically provided the experimental data, and Madhavan here at Harvard. So what we, in the general sense, what we are trying to do here is try to identify spatiotemporal coherence during morphogenesis. And this is general, can involve embryonic development on animal morphogenesis, organoid morphogenesis, cancer morphogenesis, and perhaps to be a little more specific, one of the examples that I will be covering today is about the formation of primitive streak in chick development, where basically we have thousands or hundreds of thousands of cells that at some point they will be forming the primitive streak that then will lead to the formation of the germ layers. And the question we want to try to ask is when actually do cells become faded during this developmental process? When they will be aware of forming the primitive streak, the anterior or the posterior part? So to basically address this question, there are, as far as I understand, two ways. So either you look at the single cell level and you look at the mechanistic processes involved in this cell fate acquisition, or you can do more a top-down approach, where you can use cell velocities and cell trajectories that now you can measure with the confocal microscopy, and then using this kinematic approach or the kinematics of cell motion, you perhaps want to uncover what are the mechanisms that are making these cells faded, when do they become faded, and where. So when I try to look at cell motion and start doing a top-down approach, then to identify a cell in this developmental space, we can have either gene information, so with single cell RNA sequences, or cell position information. And basically the combination of these two should be enough to tell you exactly where a cell is in this developmental phase space. However, basically the information of gene expressions when you're considering all the genes or the single cell basically transcriptomics is not as available as cell positions and not as defined in space and time. So in this talk, we want to use only cell positions and then try to ask the question that we were asking before, slightly differently, which is there a footprint of cell fate acquisition in cell position data? So we'll be focusing mainly on using cell position. So an important distinction, if you look at the kinematics of dynamical systems, is using Eulerian technique or Lagrangian, or if you will, with memory analysis. And maybe to show this difference, I'd like to show a simple example. So you may think that this is the measured velocity field that you can image when you're doing confocal microscopy and particle image velocimetry. And you have three snapshots of the velocity field, which is showing basically these three different sets of velocity field at different times. And if you just look at this Eulerian view of the process, you may think, okay, these are basically vortex type structures where you expect that cells will be circulating without much stretching. However, if you release Lagrangian particles, which I'm just marking here in green here, you can see basically that these cells will indeed rotate, but will also undergo exponential stretching. So this is basically to say that, and this is basically what I can show in terms of the movie or the evolution of cell positions that are obeying the velocity field in the background. So you can see that if you just look at the Eulerian view, you may sort of get misled of what's going to be actually the morphogenetic process that is described by this velocity field. And the other important distinction is that if you would be averaging any Eulerian quantity, for instance, the highest eigenvalue of the rate of strain tensor, which is a very typical diagnostic to measure stretching rate. So if you would be averaging that quantity at a fixed Eulerian position, say the initial position of this cell, basically that average would be completely agnostic to the path that a cell starting at this position underwent into this during this time interval. So that's why we want to identify and define everything from a Lagrangian point of view, because this is a most natural reference frame, if you will, to study a morphogenetic process. Okay, so when we want to do a Lagrangian description of a dynamical system, what we define being the dynamic morphoskeleton is a set of attracting and repelling Lagrangian coherent structures, where I'm just illustrating them here. So you consider T0 being the initial time of your experiment, and then you will start interrogating your experimental data set at different time scales. Say, for instance, you can interrogate it after 1 hour, 2 hours, 3 hours, 10 hours, right? And for every time scale, you can ask the question that, okay, instead of really chasing each single cell that will undergo a complicated path or even chaotic path, I want to instead identify the attractors and the repellers over these time scales. And what is the influence of these attractors and repellers on cell trajectories is basically depicted here. So if I have a set of cells, I don't really care about their single complex path, but I know that if there is going to be a nearby attractor over this time scale, this cell will undergo and will just start collapsing on the attractor. Similarly, I can identify a repeller that is completely basically unexplored, especially in the morphogenesis community, which is, if there is going to be a repeller of this time over these time scales, I know immediately that cells that will be starting at different sides of this repeller, they will go far away just by definition of the existence of the repeller. And now what we want to do, we want to identify the attractors and the repellers to basically describe our developmental process and where they form, when they disappear. Okay. So to identify these attractors and repellers, we use the technique that is basically standard in dynamical system, which is called finite time Lyapunov exponent. The details are not important, but the important message is the following. So what the finite time Lyapunov exponent is doing is the following. Imagine that you are in this region of the embryo, so this red dot here. This cell basically is going to have at the initial time t0 a set of neighboring cells. And then over a finite time interval, t minus t0, so the time scale that I was basically describing before, there is going to be one neighboring cell that is going to undergo a maximum separation compared to the base point. And the difference or the ratio between this maximum distance over the initial distance is basically the FTLE. So this is basically a rescaled version of the FTLE. So you can imagine that this FTLE is going to tell you a scalar field that depends on where you start, a time scale, T1, and it's going to tell that if there is going to be a region with high FTLE values, that means that cells that will be starting at different sides of this region or this ridge of the FTLE will be then separating. So I will call this a repelling coherent structure. And similarly, if I do the same analysis in backward time, I see that backward time repulsion means forward time attraction. So I will be basically showing backward FTLE and forward FTLE and backward FTLE that are basically scalar fields that will be used to identify attractors and repeller. And it's very important to remember that these are very related to the finite time deformations of this tissue. Why? Because basically the FTLE is the eigenvalue of the Cauchy-Green strain tensor, which is basically a tensor to quantify precisely the finite time deformations. So if I now go back to the primitive streak formation, so all the data that I will be showing here are using confocal microscopy and PIV derived velocity field from the formation of primitive streak in chick development. And just to recall, we want to capture the process of primitive streak formation, and just to maybe guide intuition, we know that cells will basically converge towards the primitive streak. So it's not surprising to have the primitive streak as an attractor. And this is what I'm gonna show here. So this is our region. So it's the embryo region. So this is in micron, and the embryo size is around 3,000 micron. That's basically this width here. And the scalar field that I'm showing here is a backward time FTLE that whose I values, if you will, they are marking the presence of an attractor, and these I values are basically covered by this point that will be basically become clear very soon. So the attractor at the final time is going to basically be marking the formation of this primitive streak, and there is basically a very sharp ridge, a very high value of the backward FTLE just behind this point that I will basically show immediately. So once I know that the attractor, I want to know what is basically the domain of attraction of these attractors. So where cells that will be forming the primitive streak and will ingress in the primitive streak are coming from at earlier stage of development. And this is what I can identify. So the domain of attractor of this attractor is this so-called silky region that basically marks the precise position or earlier position of cells that will be then ingressing into the primitive streak. So this is basically this region here. And how I did color code these cells that will be forming the anterior part and the posterior part of the primitive streak, just using the forward time FTLE or the repellers. So if I do the forward time analysis, the FTLE is telling me that there are going to be two repellers. So the first repeller is just dividing embryonic region with extra embryonic region. And there's going to be another repeller, repeller number 2, that is basically sharply dividing this silky region, which is the domain of attractor of the primitive streak, and is telling me that there are two distinct regions, one on one side of the domain of attraction and one on the other side of the domain of attraction. And when I color code these cells, just using the kinematics of tissue motion and deformation, then I can completely see that this repeller is identifying a footprint of cell fate specification between anterior part and the posterior part of the primitive streak, as you can basically see just by advecting these cells. And again, these are all these analysis is model independent because I'm just using experimental velocity field and then applying the technique of the dynamic morphoskeleton. So the interesting question is then when actually these repellers and attractors form. Because if I know when they form and where they form, then I can perhaps get an insight from just cell velocity or cell trajectories of when hidden morphogenetic mechanisms are making this cell fading during development. And this is what I'm going to show you. I'm going to play a movie where I show the attractors at the final time. Again, high value of the scalar field here just marks the presence or the formation of a repeller. High values here, they mark the corresponding initial position of cells that will land into this repeller. And high values here corresponds to regions that are the repellers at the initial time. So for instance, if I focus myself on the repeller number 2, this was the one that was distinguishing between anterior part and posterior part of the primitive streak. Then I see that around 300 minutes, just using the kinematics of tissue deformation, I know that cells are aware that they will be forming the anterior part and the posterior part of the primitive streak. And we can basically do the same analysis and identify when the attractor is emerging. At much earlier stage, cells are already aware that will be forming the primitive streak because the attractor, which is in the orangey regions here, is emerging with a specific time scale. And if you do the same basically analysis just using conventional techniques, for instance, the average velocity field or the Lagrangian particles position or cell positions or the deformation of a Lagrangian grid, basically you can see that you cannot really identify the formation of repellers. There is just no footprint of these quantities or this information into these conventional tools. The other thing I wanted to show is, okay, so far we analyzed basically a wild type data set or a wild type embryo. What happens if we study, for instance, an embryo which is treated? The treatment is basically the FGF receptor inhibition. So if I do this treatment, the only thing that I see basically is that the morphogenetic features are smaller. So these are the same scales. The repeller number 1, which is dividing the embryonic area with the extra embryonic area, is smaller. The primitive streak doesn't form completely, and therefore also its domain of attraction doesn't form completely. But strikingly, basically, the repeller number 2 is completely hidden. So in this treated embryo with FGF receptor inhibitor, the repeller number 2 is completely not there. So just to summarize, basically, if we know when and where the repeller number 1, number 2, and the attractor forms, we basically have a clear picture, at least from a kinematics of cell motion and tissue deformation of this developmental process. And this is also telling us that perhaps since repeller number 2 doesn't form in an embryo which is different phenotype with FGF receptor inhibition, that basically suggests that this FGF is also down-regulating perhaps the genes that are responsible for fating the anterior part and the posterior part of the primitive strict, which are basically already well known. And this is something we can sort of infer just using the kinematics of cell motion, and again, parameter-free and model-free. I just want to flash some other properties that I don't have time here to cover, but we identified also aggregate metrics, so scalars that are able to identify different phenotypes as a function of the developmental process or the developmental time. We can identify also the isotropic versus anisotropic Lagrangian deformation, and therefore distinguish the amount of cell intercalation or shear type deformation in a tissue, or cell division and cell growth. We can also identify automatically different morphogenetic features, so we can extract the primitive strict shape length as a function of time. We can extract its domain of attraction and then study perhaps the characteristic of this. And we can also identify all these quantities using noisy data. So we can completely take the data set from light sheet microscopy and PIV, and without doing any filtering, basically we can identify and recover the same features. And this is because the dynamic morphoskeleton is made of structures that are robust, structurally stable or robust. So therefore, if I start adding noise to my system, which is always the case in experimental data, those are the structures that will disappear the last. Basically, you can add the most amount of noise and still get this coherent organizer of complex motion. The second thing I wanted to show is a similar analysis that we did on Drosophila gastrulation. So this is again a different embryo where we are considering the full embryo. The motion in the early stage of gastrulation is happening on a curved surface. So you have to account properly for the curvature of this surface to identify the deformation properly. And so on the left-hand side here, I'm showing the repellers. So, and there are going to be, of course, different regions or different views. So in the ventral side, I'm going to have a very diffused repeller. This is basically the footprint of the germ band extension. So this is going to induce basically a deformation or repulsion, or a diffused basically deformation in this direction. There's going to be a repeller in the dorsal side, also diffused, because basically at the initial time cells will go from dorsal to ventral to then ingress into the ventral furrow. And on the lateral side, there is basically also a repeller due to the germ band extension. And similarly, on the right, I'm showing the attractor, which in this case, for early stage of this developmental process, is just the ventral furrow. So the ventral furrow plays the same role of the primitive streak in cheek. And what I wanted to emphasize here is that the twist gene is very well known to be expressed in this ventral region, and is very responsible for the formation of the ventral furrow. Okay, so if I do the same analysis for a twist mutant, what happens is that, of course, there is not going to be any attractor corresponding to the ventral furrow, as is very well known in this community, because twist, not expressing twist, prevents the formation of the ventral furrow. There is going to be a clear distinction also in terms of the shape of the repeller between the wild type and the twist mutant. However, there are going to be also several other differences in regions that are very far from where twist is expressed. So basically, I want to emphasize that twist is just a local difference between the genetic expression of a wild type that has the twist expressed, and the twist mutant is just the same as the wild type, but where twist gene is not getting expressed. However, this basically local change in gene expression of twist is inducing global changes captured by this dynamic morphoskeleton. For instance, if we consider the dorsal pole, which is basically here far away from the twist gene, there is basically the repeller is gone. So there is no repeller anymore. Similarly, the repeller on the dorsal, on the posterior pole is also gone for this twist mutant. However, if we would look just at the Eulerian diagnostics or tools to compare these different embryos, and we do basically detailed comparison in this reference, we see that the only difference between Eulerian techniques is just here where the twist gene is expressed. So the velocity field looks pretty much the same in all other places except where twist is expressed. So we can actually theoretically also explain why a local change can induce a global effect if you use a Lagrangian technique to capture this effect. And the reason is very simple, which is if you have a local change in the velocity field, this local change in the velocity field due, for instance, to the inhibition of twist or without allowing the twist gene to express, is going to induce a local change in the Eulerian rate of strain tensor. And this is what existing techniques would capture in principle. However, if you have a local change in the rate of strain tensor in space and time, locally in space and time, this is still going to induce a global change in the Cauchy-Green strain tensor through this exact formula, because basically you're integrating all along trajectories, basically the deformation, the Eulerian deformation described by the rate of strain tensor. So this intrinsic definition of dynamic morphoskeleton is basically is making possible to capture global effects just induced by local changes, which is basically the hallmark, I guess, of morphogenetic processes. Maybe I want to cover in the last slide one ongoing project, which is related to, so what can we really predict using the dynamic morphoskeleton? And to do so, basically, we teamed up with a group at the Olivier Pourquier group at the medical school, and they wanted to understand when the lateral plate cells in cheek development will become committed or faded. What we did basically in this schematic, we took the velocity field of cell with light sheet microscopy. You can do PIV and get the velocity field of cell during development at different stages in cheek development. And then we found basically that in this posterior part of the embryo, there is going to be the formation of two repellers around stage 7. Which is 300 minutes from stage 5. And then we were hypothesizing or conjecturing that this formation of the repeller, number 1 and number 2, repeller 2 and 3, basically were compartmentalizing this region of the embryo. And then therefore, we were suggesting that this may be a footprint or readout of cell fate specification. So what they did basically, they released a bunch of single cells. They tracked these single cell trajectories before the repellers are formed. So where there are no boundaries at stage 5. So if you release a bunch of cells at stage 5 in these regions where the repellers are not yet formed, they show that at a stage that are much later, this initial set of cells, that have basically they've been labeled, they will basically give rise to different tissues. They will basically give rise to neural tube, somites, and lateral plate. However, just doing analysis after the repellers are formed, so they released a bunch of cells between these two repellers. They tracked these cells at much later stage, and they found indeed that these repellers were identifying the cell fate specification of this lateral plate, because cells that were starting in between these two repellers were indeed giving rise only to the lateral plates and not to somites and neural tube. So this is basically suggesting that you can sometimes basically identify the readout of cell fate specification from position data using the dynamic morphoskeleton. We also did comparison and show that if you look at classical gene expression profiles at these stages, at stage 7, you cannot really identify this sharp boundary. And this therefore basically reiterates that tissue deformations and the mechanics of tissue during development is important. Otherwise, everything would be just predetermined by the gene expression information. So I'd just like to close with the summary slide that I will just leave here for you. And also I'd like to acknowledge the funding agencies that supported my postdoctoral studies. Thank you.
Thanks, Mattia. Round of applause if anyone wants to.
Yeah.
Okay. We've got time for maybe 1 or 2 questions. Okay, so we've got a 3-part question here from Ragavan. So we'll do this, and then maybe if any others can be answered in the chat. So Ragavan is asking, so number 1 is how precise is FTLE in identifying the influence of nearby trajectories in space?
I think it's as precise as the data that you get. Say, for instance, in the chick development. So this is data coming from the Keys Beecher lab at University of Dundee, and the resolution of this data set is really a single cell. So they were using confocal microscopy with adjustable height. They basically were able to identify the motion of cells at the single cell resolution. Therefore, FTL is also single cell resolved. For this data set that is basically from tissue cartography and still light sheet microscopy from the Strachan lab, the data set came with the 5-cell width averaging. So of course, if the data set is coming with this resolution, you cannot resolve smaller scales, but it's basically as good as the data you have.
The second question from Raghavan is that in your chick development, you only see one repeller attractor region, and they're asking whether if you look at other developmental stages, would you be able to predict potential other regions?
Yeah, definitely. So yeah, you can basically identify all of them. In these cases, we were just focusing on this developmental phase and in this region. So this is basically, the epiblast. So the only thing that are happening at this stage are basically the formation of primitive streak, but definitely. So if you have a bigger field of view of your experiment, then you can identify all of them together, which is also the case for Drosophila. That's basically, in this case, we did basically an in toto identification of features during this first phase of restoration.
Maybe we could just quickly finish on this one. So what are the biological differences between the repeller and the attractor regions? Do you know?
Yes, so the repeller basically, this is what we are trying to answer currently. So the repellers are very interesting because it says that if a repeller is forming during development, all of a sudden, two cells that are close to each other, they have to separate. So that means that basically there is some hidden morphogenetic or mechanistic processes in the background that is manifesting into this repulsion. The attractor instead is just the opposite. Instead of having cells that will undergo different fate, is more cells that will start co-expressing genes, or we start having a more synchronized behavior that then will form the same morphogenetic feature. But this analysis is just kinematic, so it cannot really tell the biological differences, but it tells you where to measure. So you can say, oh, there is this repeller forming, and then people can do single cell experiments and identify what does it really mean to have these repellers. And then you can do genetic analysis and say, okay, why these cells are basically getting faded at this time. Yeah.
Great, thank you very much. That's great, really nice talk. If there are any other questions, pop them in the chat. I'm sure Mattia will answer you in the chat.
Definitely, thank you.
Okay, great, thanks for that. So we're gonna move on quickly. Next up, we have Jacob Notbohm. Jacob, if you wanna go ahead and start sharing your screen. So Jacob is an assistant professor in the Department of Engineering Physics at the University of Wisconsin-Madison. He received his PhD from Caltech in mechanical engineering in 2013, and then he became a postdoc at the Harvard School of Public Health with Jeff Fredberg. So Jacob and I actually overlapped for a couple years, so I'm really, really excited to be able to introduce him here. So Jacob studies mechanical properties of biological materials and how physical interactions between cells and their surroundings control cell contraction and migration. Jacob, the floor is yours.
All right, thanks. Thanks for the introduction. You can hear me okay? Give me a thumbs up, Jen. Yeah, or
yep,
not.
You're good.
Thank you. Yeah, so I'm going to talk about some of our work in collective cell migration, and some of this actually started in my postdoc at Harvard School of Public Health and Jeff Fredberg's group, and working with some other people, Jim Butler and Jenna Park and Jen Mitchell. So I'm going to start out actually with a movie taken by Jen Mitchell, working with Jenna Park. These are cells that we're looking at on the left. Well, they're primary cells, so they're from human donors on the left, from healthy donors, and on the right from people with asthma. And the cell type is from the airway. They're from the human airway, and they're cultured in an air-liquid interface to simulate those conditions as well as possible in the lab. And I'll play the movie on the left. These are from the healthy donor. The cells just kind of are stuck in place. They don't really move. Language that we would use is jammed or solid-like. Think about cars in a traffic jam not moving. And on the right, I'll play these. And these are a lot different, right? The cells are flowing, they're rearranging with their neighbors. We could call it fluid-like since we see flow. In contrast to jam, we could call them unjammed. And so you see these movies and you're thinking, okay, well, what does this have to do with asthma? How does asthma create this difference in motion? Right, but there's a more fundamental question, which is just in general, what causes differences in motion in collective cell migration? And that fundamental question, we still don't really have answers to. So we can't really address the application question to asthma yet until we get at the fundamentals. And so that's what my talk is going to be about. So what I'm going to talk about is getting at these fundamentals of how forces produce motion in these collective systems. I'm going to start out by using a model, and this model has been published in several papers. The ones listed at the bottom are a few commonly cited ones. And what the model does is it takes the sheet of cells that you see here. Here's a cartoon that is what the model is trying to simplify those cells as being. Each cell is sort of a polygon like you see on the top here. And the model looks at different components in the cells that produce force. And so thinking about actin and myosin producing contraction. Also, the cells are connected together with some cadherins. And so putting all of those together, we have to write equations for how these different molecules affect the force or the mechanics, the energies in the system. And so the equation that has been commonly used is this one here. E is an energy. It's the energy of the ith cell. I is the cell. And there are a few different terms, 3 different terms in this equation. So I'll go through those. The first term is a tendency to maintain a preferred area. And A0 is that preferred area. AI is the area of the ith cell. And so if AI and A0 match, the energy is minimized. And this term is quadratic in energy. And so that means essentially it's like a linear spring. And so there is a spring associated with the cell area. Its spring stiffness is related to this constant Ka. And so that's the first term. The second and third terms are both with perimeter. Pi is the perimeter of the ith cell. There's a term that's quadratic in perimeter. This is supposed to be something related to some active tension in the cortex. It's kind of like a spring as well, since it's quadratic in perimeter. So you can think of this term as being like a spring-like term associated with cell perimeter. And that second, the second perimeter term, the last term in the equation, is related to just the perimeter linearly. And this term is supposed to be account for cortical tension and adhesion. And so you can think of it as follows. Cortical tension would tend to contract the size of the cell. Tension around the cell perimeter would tend to make the perimeter smaller. Adhesion would do the opposite. The cell-cell adhesions are associated with, or they create a sort of a gain in energy. It makes it preferable for perimeter to be larger if you have more and more adhesion. And so this last term is supposed to represent this interaction between adhesion and cortical tension. And these equations can be simplified, or this equation can be simplified as follows. The first term is the same, and then the second term here has been rewritten to have the same type of structure as this tendency to maintain a constant area. Now it's a tendency to maintain a constant perimeter. And to get from the first equation to the second, you could see if you would expand out the squared term, that there'd be a pi squared. There'd also be a term that's linear in pi, which is this one here. And there'd be a PO squared as well. But PO is a constant. Ultimately, what we're interested in is forces. Force is a derivative of energy, a derivative of a constant is 0. And so the fact that this extra PO term has been added onto the equation doesn't really affect the results of the model. And so this simplifies understanding this model a little bit. You can just think of this tendency to maintain constant area, tendency to maintain constant perimeter. And so that's what the model does. Now we're going to look at some predictions from the model. Again, this is that same equation I showed you on the previous slide. There are a couple different constants in the model, this Ka, Kp, and the A0 and P0. It turns out one of the more important ones is to combine P0 and square root of A0 into this dimensionless term. This is like a dimensionless preferred perimeter. And so bigger P0 over square root of A0 would be a more elongated cell shape, and a smaller one would create a more circular cell shape. And what the model predicts is that this dimensionless parameter affects this transition from solid-like or jammed on the left, and that would be having a small p0 over square root of a0. And on the right is having a larger value of p over square root of a, and this is more fluid-like unjammed motion. So that's the prediction of the model. I can explain a little bit more intuition as follows. For a small perimeter square root of area, each cell is going to tend to be a circle. And the cells have to pack together. And so the only way to do that is to have circular-like cells that are all stuck in place. And there's only one really configuration of cells that minimizes that. It's a sort of honeycomb-like hexagonal structure, and that would tend to minimize cell perimeters. By contrast, if perimeter square root of area is big, cells are elongated, and there are many ways to be elongated. You could be elongated, stretched horizontally, you could be stretched vertically, you could have some curvature. And so each cell has infinite conformations it can take to achieve this preferred perimeter. And then because each cell has infinite conformations, when you pack them together, they're infinite packings. Switching between those infinite packings is relatively easy, meet low energy costs, and so you get this fluid-like behavior because of that. And so that's what the model says. And there's one more piece. It might be obvious, but I want to point it out. P0 and A0, these are inputs to the model. These are like material properties associated with the cell. And then P and A are the actual cell perimeter and area. And so when you have a smaller input, a smaller preferred perimeter over square root of area, you get a smaller actual perimeter over square root of area. And same thing when perimeter square root of area is larger, when the preferred values are larger, the actual values are larger. And so this creates a prediction that we can test in the experiments. We can measure P over square root of A in the experiments. And so going back to the intro slide showing cells from the non-asthmatic or healthy donors and the asthmatic donors, what we see is the asthmatic case where cells are more easily able to rearrange, flow past each other, more motion, more fluid-like motion, also have larger P over square root of A, which we see here. So things are consistent with the theory. And so this is all been work that was or pieces of this right here were done when I was a postdoc at Harvard School of Public Health. When I started out my own lab, what I wanted to do is figure out what is it that causes this difference in perimeter and motion. And looking at this picture here, we say, well, it's this P0 and A0, but then the question is what causes those, right? And so that's what I'm going to talk about for the rest of the talk today. So what we did is we went to a system that has been established before in the literature, and that system is the effect of density on collective migration. So we're not the first to see this effect, but we wanted to use this well-established effect to investigate our question about what causes these differences in perimeter. And so what you're looking at here, these are circular islands of cells that are about 1 millimeter in diameter. The only difference between left and right is the cell density. There's approximately 3 times the number of cells on the right. And you see very different motion. The high density case, the cells are jammed or stuck in place, solid-like. We quantified the motion. From the images, we approximately trace cell trajectories. Trajectories in the boxed regions are shown here and here. You see at low density, the long trajectories and curves, meaning cells might be rearranging or sliding past each other. And at high density, the cells are just stuck in place. They're not really moving. To quantify this, we computed mean square displacement. The equation's up here. R is the position of the ICE cell. We take the difference of position at 2 time points, so we square it and take a mean over all cells. And then we repeat that for different time differences, delta t. And so here's that delta t versus against the mean square displacement. So obviously less motion at high density. So the high density case, you see smaller mean square displacement. The slope though is also important. So I'm showing you slopes of 1 and 2 on these logarithmic graphs. Let me describe the slope a little bit more. Looking at this alpha 2. Alpha of 2 means that mean square displacement goes as time squared. And so what that would mean is displacement is linear with time. So that would be just constant velocity in a straight line, a straight trajectory. So that's kind of the upper limit of the slope. And then alpha's 1 would be consistent with a random walk, just sort of random motion. So what we see here is that at high density, the slope, alpha, is smaller, and that's consistent with this fact that there's less rearrangement, fewer cells sliding past each other. looking back now to the prediction of that theoretical model I showed you, we also measured cell perimeters. So P over square root of A, from here out, I'll use the symbol Q, and that's this dimensionless cell perimeter. At high density, the dimensionless perimeter is smaller, which is consistent with what we've seen before. All right. So what we wanted to do is figure out what causes this, right? We said it's P0 and A0, at least that's what the model predicts. And so that's going to be our hypothesis. Is what causes the differences perimeter? We'll hypothesize is this P0 over square root of A0. And so the hypothesis then is that P0 over square root of A0 affects the actual perimeter, which you saw on the previous slide, and the rearrangements also on the previous slide. And then even further is one more point, which is this preferred perimeter is controlled by things at the cell periphery, right? So actomyosin in the cortex, and I said previously that the cortical contraction would reduce the perimeter, and then adhesion, like due to cadherins, would tend to increase the perimeter. So there's a lot on this slide, but our working hypothesis is just in this box here, which is that cortical actomyosin and adhesion affect the cell perimeters, and that also they also affect the cell rearrangements. So this is our hypothesis, and I want to point out that we're not the first to propose this relationship between actomyosin and adhesion molecules, a relationship between those on cell shape and motion. There are various other citations in the literature that suggest similar types of relationships. And so this hypothesis is based on current understanding in the literature. So what we did then is we said, okay, actomyosin, adhesion, we have this system with two different densities with different perimeter and motion. So let's look at the actomyosin and the adhesion in that system. So that's what we're looking at here. These are images showing fluorescent images for actin, cadherin, myosin. This is cells at lower density and higher density. So we look at these images and we don't really see a difference, right? And we try to quantify it. And here is a quantification of fluorescent intensity of cadherin, decadherin on the junctions. No real difference. Each dot is a different cell island, so a different sample. We also looked at junctional myosin, thinking cortical tension is going to be related to the amount of myosin at the cell-cell interfaces, right? And again, no difference. And so, given this hypothesis, we expect that there'd be a difference in because there's a difference in perimeter rearrangements. We'd expect a difference in one or both of these, and we don't see one. The fluorescent imaging isn't the greatest way to quantify things. It's only semi-quantitative, and so we did another experiment where we did laser ablation to measure cortical tension. And this is a movie here. Let's see if I can play it. Uh-oh. And so we ablate a cell-cell junction, and you can see retraction. On the left is the control or lower density system, and on the right is the higher density system. We repeated this for many cell junctions, and then what we did is we plotted the distance between two vertices connecting the junction, and we measured that distance over time, subtracted the initial distance. So we plotted that against time. And you see greater slopes at low density, right? We quantified the slope by fitting to a line, which is we call the recoil velocity. And you see that the slope is higher at low density. That suggests greater tension, greater cortical tension for the system of low density compared to high. So the observation here is that when we increase the density, cortical tension has gone down and perimeter rearrangements have gone down. So you compare that to our hypothesis, it's the opposite of the prediction. Right? So density, the data is a little bit inconsistent with our hypothesis. So we moved on and we thought, let's look more directly at the molecules associated with actin and myosin contraction. And so we used inhibitors, right? So we start with blebbistatin, which inhibits myosin contraction. We imaged actin in the cortex. And what we found is that the cortex became more curvy, which might mean that each cell edge is under less tension if it's more curvy. This curvature index is a measure of the overall distance of the edge divided by the end-to-end distance. So greater curviness could mean that the cortical tension has gone down in response to the blebbistatin. We also used another drug, cytoclasin D, to disrupt actin polymerization. Here we measured phosphorylated myosin. And we saw a reduction of that in the cortex. So that also might mean reduced cortical tension. To verify these, we also did the laser ablation experiments that I previously showed you. I think we're a little short on time, so I'll just point you to the recoil velocity data, showing that both blebbistatin and the cytoclasin D reduce the recoil velocity, probably meaning reduced cortical tension. Right, and so the prediction is with reduced cortical tension, we should see an increase in perimeter and rearrangements. So here is data on perimeter and rearrangements in response to blebbistatin. We see for different concentrations, the perimeter has gone down. We also see a reduction in the rearrangements. Same trends were observed for cytoclasin D, a reduction in the dimensionless perimeter and a reduction in the rearrangements. Note though, these are opposite to the hypothesis. We've reduced cortical tension and we've reduced perimeter and rearrangements. Right? So this initial hypothesis that I've been showing you is in conflict with the data, right? So the conclusion here is something's wrong. We were wrong, we're missing something. So what we did is we went back to the model, revisited things. And the model that I showed you, here's the equation I showed you. The equations here are only related to an energy. There's no forces in this model. And also, ultimately, if you want to look at motion, we may need to think about forces, right? Forces create motion. So in a later iteration of this model, cited right here in Physical Review X, forces were added into the model as follows. The first thing is a gradient of energy is force. And so the energy term gets inserted right here. The next term I'll point to is over here. X is the position of each cell, and so its time derivative is velocity. This factor eta is like a viscosity, and so this term here is like a force as well. And then the last piece of this model is this active force. This is a force that the cell applies to propel itself forward. This force it's the traction applied by the cell to the substrate beneath. Right. So this is the new addition to the model. This force, I'm using the symbol F for the active force, right? And so then the model predicts, now there are two important variables. One is this P0 over square root of A0, which I talked about previously, and the other one is the active force F. As you increase P over square root of A, you go from a solid-like to a fluid-like system. That's what we discussed previously. The new piece is as you increase F, you also go from a solid-like to a fluid-like system. Also at the same time, perimeter over square root of area increases. The actual perimeter over square root of area increases in response to increasing either of these two factors. All right. So that's the prediction. This slide is essentially a repeat of the previous slide, but it's important, so I'm just going to repeat it again. That in adding in this force equation, there are two different factors that could cause perimeter over square root of area to increase, or and motion and the motion to increase. The first term is this one, which we've already sort of evaluated. We've looked at actomyosin contraction in the cortex. We looked at cadhereins in the cortex. Didn't really see much. We didn't really see anything consistent with what would cause reduced rearrangements. So that suggests this new term F might be important. So here's the new hypothesis. New hypothesis, this force F is the traction applied by cells to substrate, and that is what's responsible for the changes in perimeter and rearrangements. So to test this out, we'll measure tractions. This is forces applied by cell to substrate. We use traction force microscopy. These are the data at different densities. You see at higher density, it looks like tractions are reduced. This data here quantifies a root mean square of traction. The control case or the low density case, the tractions are higher compared to high density. This is consistent with this idea that reducing forces would reduce the motion, or increasing forces would increase the motion. There's a little subtle issue here related to tractions, that the previous slide showed a root mean square, but in the equation, this force F is a vector. And the root mean square is not necessarily the same, because it could be that greater root mean square traction is caused by a bigger force produced by each cell, but that cell itself is in force equilibrium. And what the model accounts for, this F is related to the net force that the cell applies. And so if that net force of the cell itself is in equilibrium, then that net force could be zero and F would be zero. And so we had to do some analysis to get at this imbalance of forces. I was told by Jen that we're a little short on time, so I'm actually going to skip the rest of this slide for time. Details are in our paper. The conclusion of this slide, though, is that the trends with root mean square and the force imbalance analysis that we do are the same, right? So I'm going to use the root mean square for the rest of this talk, with some understanding that the trends are the same as looking at like this imbalance issue and thinking about forces more as a vector instead of an average scalar. All right, so here are data of cell substrate tractions. I've already showed you the effect of the density. This is the effect of lovastatin and cytokinase inhibitors. As expected, they reduce the tractions. These are data quantifying that reduction, right? Previously, we've seen that reducing tractions reduces perimeter. Previously, we've seen that the lovastatin and cyto-D reduce perimeter, reduce rearrangements. Here we see, as expected, they reduce traction. So these data are all consistent with our new hypothesis that I'm showing here. The thing is, all of these data reduce. The hypothesis could also be that if we increase traction, we should increase perimeter and rearrangements. So what we sought out to do is figure out how can we increase traction. So a few things we thought of. One was an activator of Rho, CNO3. This increases the tractions. The quantification of that is shown here. This is tractions over time for different cell islands. The CNO3 is shown in red, and you can see that tractions increase greatly over a period of a few hours. Compared to control, the average increase is about a factor of 3. All right, and so this Rho activator increases tractions. The prediction now is that we should increase perimeter and rearrangements. So here's the data of perimeter. Our baseline case versus our treated case, we see an increase in the dimensionless perimeter as expected. There's one more note here. Notice I'm labeling these high. These are cells at higher density. This is data for cells at lower density. And so what you see here is that the treatment with the CNO3 has sort of reversed the effect of density. These two groups here are statistically different. These two groups, the low and the treated, are not different. And so what we've done here is we've reversed this effect of density on the cell shapes, on the dimensionless perimeter. Here are data for mean square displacements, looking at rearrangements. This plot's a little bit messy. It's easier just to go straight to the exponent, the slope of mean square displacement over time. And we see, again, the effect of the CNO3 increases the rearrangements as compared to just the high density islands. These data are statistically different. And we've also almost gotten an increase to the point of the low density. So we've partially reversed the effect of the density on the collective motion. All right, so all of these data are now consistent with this new hypothesis. This here is just a movie showing motion and shape in the control case and in the treated case. And we see long trajectories, curved trajectories in this treated case, indicating fluid-like motion. If you look carefully, also you see the cells are more elongated. All right, so now there's a little bit of an issue that somebody brought up. This was during the review stage, and it's, well, how do you know that the tractions are the underlying cause? Maybe there's some other underlying cause. So for example, maybe it's that the cell perimeter is increased and then that causes traction to go up. And so what actually the reviewer suggested is like you should look at the timing of this, and you should see that tractions are going to, if you're correct, traction should increase first. And so that's what we did. So this is showing traction over time for one of our experiments, showing high density, and one experiment with the treated case. The treatments applied at time 0, and these dotted lines here correspond with different snapshots that you see across the bottom. And again, high density is here, and then this is the treated case. So looking first at traction, we see after maybe a couple hours, traction's increased. But if you look, it looks like the increase in the cell perimeter, you don't really observe that until maybe this 180 minutes or 230 minutes, right? So my student Ashwin was showing me this data, and I said, yeah, maybe you're right, but looking at these images is qualitative. maybe we're just seeing, a trend that we want to see, namely that, traction increases around 120 minutes and the perimeter at 180. So maybe we just want to see that. So I said, print out these 5 pictures and show them to different people in the department and ask them which cells are more aligned than others, right? So he did that. And he went to a group that studies computational fluid dynamics, and they look at meshes of grid points, and they look at how those meshes deform as a measure of how the flow fields are evolving in time. And that group said, oh yeah, it's obvious, 180 minutes, this image right here, the cells are becoming more elongated, whereas these two are no different. And so that to me was pretty convincing, because this group is experts at looking at meshes and grids like this, and they're very certain that the elongation occurs at this time point. But in case you're still not convinced, we did some more quantification, which we show here. This is traction over time, and this is dimensionless perimeter over time. Data shaded in gray show when traction is statistically different than the baseline, or when perimeter is statistically different from the baseline. So what we see here is traction takes about 2 hours, 120 minutes after the treatment to increase. However, the perimeter takes longer. In our data, it takes about 230 minutes. And so traction increases before perimeter. That suggests some causality, that the tractions are the underlying factor that affect perimeter and also the collective motion. There's one more point I want to get at, which is thinking about what structures in the cell create traction. And it's these stress fibers, right? These are images taken at the base, the bottom of the cell, showing this well-developed actomyosin stress fibers. These images, the image on the left is for cells at lower density, where traction is high. The image at the right here is at higher density when traction is lower. And so we see fewer stress fibers, and that's quantified in this graph here, showing an order parameter. The order parameter is describing alignment of the stress fibers in each cell. And so a bigger order parameter would be more alignment, more developed, more organized stress fibers. And so we see that at lower density, right? So it could be these stress fibers because they have this aligned structure that those affect the perimeter, right? And then that the underlying factor affecting perimeter is really the stress fibers, not the traction. It's possible, right? We began to investigate that a little bit. We disrupted stress fibers with this inhibitor of formins. And formins are needed for establishing the stress fibers. Clearly no stress fibers in the inhibited image. This is effects of this inhibitor on the perimeter. They cause a decrease in perimeter. And so this is suggesting that stress fibers could be involved, right? We can't really separate out whether it's tractions or stress fibers that affect the perimeter. Looking back at the theory, the theoretical model actually isn't very helpful to answer the question of stress fibers versus traction, because the equations shown here, starting with this term on the right, this is looking at things related to the periphery, the perimeter of the cell. Stress fibers are inside the cell, and so this term's not related to stress fibers. The first term is related to the area, which is inside the cell, but this term is just isotropic. It's just area. It has nothing to do with any elongation. And so the model doesn't have any anisotropy due to stress fibers built into it. So we really can't answer the question of whether stress fibers or traction are really the predominant underlying factor. So as I said, it's possible that the greater perimeter is caused by the stress fibers, right? But it's also possible that it's caused by traction, right? And our justification or reasoning for that is if you look at this model, the results of this model, increased traction both increased the amount of motion from a solid-like to a fluid-like motion, but it also increased the perimeter, the elongation of the cells. And that you can see here, in this fluid-like case, the cells have slightly more elongated shapes, right? And so we can't really answer between these two, but it's possible that it is indeed it is traction. It's possible. All right, so that's it. I'm just going to summarize the talk here. Starting at our initial hypothesis. The initial hypothesis was looking at this axis of this phase diagram, looking at preferred perimeter, and we thought molecules in the periphery, cortical actomyosin, adhesions are what affect the perimeter and the rearrangements. But what we found is really more related to this axis here, traction magnitude. Bigger tractions give you bigger perimeter and bigger motion, more motion, cells sliding past each other, rearranging. We also showed that tractions reverse this well-established effect of density in collective migration. And so I just want to conclude with one more point, which is we're not refuting this notion of perimeter being important. All we're saying is when traction is large, it seems to dominate over the effects of perimeter. That's what we're suggesting. So built into this argument that we're making, this conclusion that we have, is that this conclusion requires there to be tractions that are large. So there could be some systems where tractions are small. And in those systems, our hypothesis or our conclusion would not necessarily apply. So things like maybe embryonic development, maybe collective motion in a cell spheroid. Cases like that, perhaps tractions wouldn't be as big, and the preferred perimeter would be the key factor. So that is it. I just want to acknowledge Asha Saraswathibhatla, who did the experiments that you saw here, and thank you all for your attention.
Thank you, Jacob. If people want to unmute their microphones and clap, thank Jacob for the great talk. So did I miss it right at the end before we launch into other questions? For the formin inhibitor, did you also do the traction experiments for that?
Yeah, we did. I didn't show it here, but yeah, tractions were reduced by the formin inhibitor.
Okay, so that also did what you expected.
Yeah. Mm-hmm.
Yeah, our first question is from Raghavan, who asks, what happens when cell-cell adhesion is reduced? Can this somehow replace the substrate friction? For example, in a monolayer with less substrate interaction or a suspended monolayer, less substrate friction could also lead to less stressed fibers.
Yeah, okay. So I guess there's two questions built in one. One is looking at reducing cell-cell adhesion. The other is if you suspend the monolayer. So I guess let me talk about the second one first. I think if you could suspend the monolayer and have it be relatively stable, then there are no tractions as you described, and the effect of cell shape and motion would primarily result from cell-cell adhesion and cortical tension, some balance of those. So I think that is what would occur. And there's some work in embryonic development showing that type of thinking is correct. Showing if you look at, say, fluorescent label myosin, you look at that in an embryo, you can correlate some of the myosin to some of the myosin on the cortex to collective rearrangements and things like that. The third, though, is what happens if you alter the cell-cell adhesion. We looked into that a little bit. It's a little bit difficult to alter it with just some type of quick chemical factor. You'd have to perhaps do some transfection, which is a little trickier, just more time-consuming. And so we don't have a firm answer on that. There's been some other people doing work in that area. An additional difficulty is if you say affect the cadherins, you're also going to affect the cortical tension at the same time. So I think there are other people that are doing work along those lines. I just don't have anything specific to discuss from our work on it.
Thanks. We've got a question from Leon Rossetti, who's asking if you could use local optogenetic row activation to create regions of unjammed within a jammed monolayer.
Yeah, that would be cool. I think so. I've seen some of that optogenetic stuff. It seems to work pretty well. I've never used it, so I don't know if it would be finicky. In principle, it should work. So yeah, try it out. Somebody try it, and
I think
this discussion when you do it and publish it.
I think Karen Kaska's group at Columbia is using optogenetic tools and investigating some of these questions about the different how cell mechanics and cell rearrangements are happening. So
yeah, she's doing embryos though, right? So that would be
embryos, yeah.
Optogenetic expression of the or activation of cortical tension it'd be cool to do a comparison then of traction, try to do the same thing on traction. And, you know, in our system, we have large tractions, and so we think that traction axis is important, and so we check that. And then in Karen Kaye's system, she'd look at that other axis of the phase diagram and see that. And so that'd be cool. That seems like multiple papers. So if somebody has those optogenetic tools readily available and wants to talk to me, look us up. Be happy to talk.
Nice. Okay, great. We have a question from Gareth Jones, who says, asthmatic cells are more mobile and monolayers. So is this due to reduced cell density, increased row activity, or both?
So I didn't show it at the start of the talk, but tractions applied by asthmatic cells were higher than normal. So I think a lot of it is caused by increased forces. But there's some other complications as well. And Jen Mitchell has a paper on bioRxiv that is going to probably be the best answer to that question.
Thanks. Soon to be published.
Next question's from Amrinda Nain, who says, nice to talk, Jacob. How does the total distance traveled change with PO over A to the root of A number? Do you observe any sudden jump at a value of this number?
Yeah, that's a good question. So let me say something that I think what you're getting at, which is that this P over square root of A, the theory predicts there's a critical value where you get a phase transition from solid to fluid. Our data, we never get to that critical value. So in our experiments, the actual P over square root of A is always bigger than this sort of critical value. And so that kind of creates a little bit of a complication because we don't have a discrete phase transition. So that is, one comment. But your question was about total distance traveled by the cell. And it's something I don't think we even ever measured it. So I'm not sure, I can't answer directly, but I can say just looking at the pictures of the trajectories in the cells that we call more fluid-like or more rearranging, they are moving with a larger distance. We just never quantified that.
Yeah, I agree. I think they'd definitely, if you did quantify it, they'd be moving more. Venkat asks, says, great talk. Do you think the basal area and hence the energy of integrin ECM adhesion matters?
I would say yes, sort of. I don't know about area. So area is coupled to the amount of force that the cells apply, apparently, right? You saw at higher density, the cells have a smaller area, and the force goes down. And so there's some type of active mechanosensory, something that causes the cells to contract less when the area is smaller. But I guess your question is more broader about like the cell substrate energy, which in a way we've kind of gotten at by looking at the traction, right? The traction's at cell and substrate, and we've shown when that is large, you get more motion, you have a larger perimeter. And so the answer to your question, I think, is yes. I would generally agree with some of your thinking that like the energies associated with cell substrate interaction in our system are dominating the energies of the cell-cell interactions.
Maybe just a quick question from me. So when you increase the traction, you're causing this more seemingly fluid, or at least the stress fibers, we don't know if it's traction or not. How are those tractions oriented within a monolayer? I mean, how does that process happen?
Tractions oriented. So we're kind of just getting into that now. So we're interested in the traction applied by each cell. Right? It's a little tricky because we can measure tractions and our spatial resolution is good enough, but we also have to be able to segment the cell and identify its boundary, and that's kind of tricky. Just you need good quality images to do it. So I don't have a good answer yet, other than to say it's something we're thinking about and working on. We're now able to, Asher has a method where he's able to take the vector sum of traction inside each cell, which we weren't able to do when we were working on this paper. How that vector sum on a single cell level is affected by the activator, we haven't yet quantified. We've been looking at more like changes in time of a single cell and not looking at comparison between activated or unactivated. But it's something we could look at. I think Asher's watching, so Asher, write that down.
I guess the difficulty is in a monolayer, the cell could be exerting a traction which affects something on the substrate in the cell in front, and you don't know how far that effect exists, right?
Correct, although we are now able to look into it.
Okay.
Yeah.
I think there's work out there, right? That's sort of, I don't know what the numbers are, but that can tell us like how far deformations can propagate within the monolayer, or sorry, within the, through the substrate itself, if that's what you're getting at, Adam.
Yeah, I mean, how far in front would it exert? I mean, is the traction, I mean, there'll be a local area, right?
So I guess I was thinking about, like, you were asking about coordination of cell attraction between cells. Like if, one cell applies traction, is somehow another cell in front of it applies similar traction. Is that what you're asking?
Yeah, that's what I meant before. Yeah.
Yeah. So like Asher's kind of looking at those types of things and looking at like correlations in traction between one cell and its neighbor. And over what distance that correlation occurs, and is it more like front back or left right, or makes no difference. He's looking into those types of things now.
All right, great. I think that was our last question. So I'm going to go ahead and thank both of our speakers, Jacob and Mattia, and I'm going to stop sharing to you.